Hi, this lecture is going to explain nerve conduction, which includes action potential initiation and conduction of action potentials as well as graded potentials. So in a sense, this lecture is going to be part 2 of our nervous tissues chapter. So hopefully you've listened to part 1 of the nervous tissues chapter, which talks about the structure of the neuron, the different types of neuroglia, and fast and slow axonal transport. So I'm going to start this part by quickly giving a refresher about the structure of a typical neuron. So to recap, this is what a multipolar neuron looks like. And these are the most abundant type of neurons. So this is the body of a neuron, which is referred to as a cell body. A lot of times it's called the cyton or the soma as well. So the soma is the part of the neuron where you have the nucleus, you have the rough endoplasmic reticulum, smooth endoplasmic reticulum, the lysosomes, the mitochondria, all your typical eukaryotic cell organelles. Now, in a multipolar neuron, you have a lot of different projections or dendrites that originate from the cell body. In a unipolar neuron, you only have one projection exiting out of the cell body. In a bipolar neuron, you have two projections exiting out of the cell body. So that is what makes this neuron a multipolar neuron. So these projections, the short projections sticking out of the cell body or the soma, are called dendrites. The longest projection that sticks out of the cell body, this one in this instance, is called the axon. So the dendrites basically serve as antennae. So these dendrites could synapse or join with other neurons, or they could synapse with sensory receptors, or they could synapse with muscle tissues. It could be anything. So that is what makes a neuron a sensory neuron if they are synapsing with sensory receptors. If they are synapsing with muscle tissues, it would make them motor neurons. Or if they're synapsing with other neurons, it could make them interneurons. Now, the axon could be either myelinated or it could be unmyelinated. This particular neuron in this illustration has a myelin sheath. So that is what makes this neuron a myelinated neuron. So what myelin is it's a lipid-based layer that keeps the nerve impulses from leaking out. So it insulates the nerve impulses. It speeds up the conduction of nerve impulses as well. How it speeds up the conduction of nerve impulses is something we're going to talk about a little bit later during this PowerPoint. Now, the myelin sheath is not continuous. It has little gaps at regular intervals. You'll see these regular gaps right here. And those gaps are referred to as nodes of Ranvier. So we've talked about the dendrites, we've talked about the cell body, we've talked about the nucleus as well. So this illustration is only illustrating the nucleus, but other cell organelles are also present in the soma or the cell body. Now, right here, we are going to assume that this is a neuron in the peripheral nervous system. You have support cells that synthesize the myelin sheath. They are called Schwann cells. So Schwann cells are myelin synthesizing cells in the peripheral nervous system. If this was a neuron in the central nervous system, the myelin synthesizing cells would be oligodendrocytes. Now, at the end of the axon, you have several terminal dendrites. So these are all terminal dendrites. And at these terminal dendrites, you have axon terminals. This is an axon terminals, this is an axon terminal, this is an axon terminal. And at each axon terminal, you'll see a little bump at the end. So here's a bump, here's a bump, here's a bump. So the bumps at the ends are called the synaptic end bulbs. So the bumps are called the synaptic end bulbs. Now the synaptic end bulbs may also synapse with muscle tissues, they may synapse with other neurons, or they may synapse with sensory receptors. So we'll talk about what happens at a synapse a little bit later. You might remember this from our neuromuscular junctions from our muscle tissues unit. But something very similar happens between a neuron-neuron synapse as well. And we'll talk about it a little bit later during this PowerPoint. But in order to understand how an action potential or a graded potential is generated, we talked a little bit about action potentials versus graded potentials in the previous part of this lecture. We need to understand what resting membrane potential is. So a graded potential is basically an electrical impulse that travels a short distance. So a graded potential is an electrical impulse that goes over the dendrite and over the cell body. So it takes this path. So it goes a short distance over the dendrite and over the cell body. Now, you are exposed to various stimuli all the time. Not every stimulus generates a response. If the stimulus is large enough to generate a response, then a long distance response is generated. And that long distance response is called an action potential. So if a graded potential decides to turn into an action potential, which happens at this junction. You guys already know what this junction is called. It is called the axon hillock. Now, the axon hillock is the junction where a graded potential is going to turn into an action potential. So if it is deemed worthy of being converted into an action potential, this is where it's going to happen. And an action potential is going to travel all the way across the axon. It is going to travel through the axon terminal. It is going to go to the synaptic end bulb. It is going to jump over to the synapse and go to the next neuron as well. So we're going to see how that process is going to take place. But in order to understand how that process is going to take place, we need to understand the concept of resting membrane potential. So let's look at what resting membrane potential is. Now here is a zoomed in image of the axoplasm and the axolemma, which is nothing but the plasma membrane of the axon or the neuron, whatever you want to call it. So here is the plasma membrane of the neuron. It's a lipid bilayer. You have these transmembrane proteins right here. So in this case, they're serving as ion channels. This is serving as a potassium ion channel. This is serving as a sodium ion channel. The peach shaded area is the inside of the neuron, which is the cytoplasm. The blue shaded area is the extracellular fluid, so outside of the neuron. This blue transmembrane protein here is a very important transmembrane protein. It is the sodium-potassium ATP synthase, the sodium-potassium pump. Now, if you were to stick an electrode, which is nothing but a needle that is inserted into the plasma membrane and connected to a voltage meter, you could read the voltage. In this case, it is in millivolts. So you'll see this red arrow is pointing towards the millivolts. In this case, it is going from the negative 70 to 0 to into the positive scale. So needle is inserted, it's connected to the electrodes, it's reading what the voltage in millivolts is. Now the inside of the neuron has more potassium ions, which is K+. The outside of the neuron, aka the extracellular fluid, has more sodium or Na+ ions and less potassium ions. Inside you have more potassium and less sodium. However, the inside is more negative because you have more proteins. I'm sorry for my bad handwriting here. I'm trying to write with my mouse. The inside is more negative because you have proteins which carry a negative charge. You have DNA which carries negative charge. So the net charge on the inside of the neuron or the cytoplasm is negative because you have these large macromolecules on the inside which carry a net negative charge. The outside is more positive because the outside doesn't have these large macromolecules which carry a negative charge. Hence, the outside is largely positive because all it has is these sodium ions, which are positive. So if this needle was to measure the charge inside, it is measuring negative 70 millivolts. So the resting membrane potential, therefore, in a neuron is negative 70 millivolts. When the neuron is at rest, there is no stimulus going on. When the neuron is at rest, the resting membrane potential, therefore, is negative 70 millivolts. And the explanation for that, why it's negative on the outside versus why it's positive on the outside, is because DNA and proteins and all the large macromolecules inside the cell has a negative charge versus on the outside there is nothing big that carries a negative charge. Hence, the outside is more positive and the inside is more negative. Now, the sodium ions cannot come inside. One, they cannot just diffuse through the plasma membrane. They cannot come inside through the sodium ion channels because the sodium channel here is closed. The potassium from the inside cannot go outside because it also cannot diffuse freely through the plasma membrane. And the potassium gate here is also closed. So sodium cannot come inside because the gate is closed. Potassium cannot go outside because the gate is closed. The only way they can go in and out is if the gates were opened. And for the gates to open, something has to happen. That something is going to be a stimulus. That stimulus could be a sound, that stimulus could be a taste, that stimulus could be an itching sensation, it could be pain, it could be wind on your face, it could be music, it could be a scream, it could be anything. So there has to be a stimulus of some kind. So in picture B, which is the picture lower, this image right here, a stimulus has taken place. Now, when the stimulus took place, your sensory receptor, so let's say there is a pin on the desk and you touch a pin. Touching a pin is not a pleasant sensation. So when you touch a pin, your sensory receptor detected a change, an unpleasant change. So in the neuron, as a result of detecting that change, the sodium gate ended up opening. When the sodium gate ended up opening, what is going to happen? Naturally, the sodium ions are going to gush inside of the cell. When the sodium ion gushes inside of the cell, now you have suddenly this influx of positivity going inside the cell. So look at what is happening to your charge. So look at the voltage meter here. So from negative 70, it's going towards 0, positivity is increasing, and the positivity is going to keep increasing until the sodium gates are open. So until the sodium gates are open, the positive charge is going to keep on increasing and keep on increasing and keep on increasing. Now, at some point, sodium comes in and the sodium gate is going to shut off. So temporarily, what has happened is that on the inside, you have a positive charge, and temporarily, the outside has become more negative than the inside. Temporarily, of course. So the inside has become more positive, the outside has become more negative. Now, this is not normal for a neuron. This is not normal for a neuron. Now, in order to revert back to its normal state, the neuron has to somehow kick back the positive back to the outside. And in order to do that, it is going to end up opening the potassium gates. Now, as the potassium gates open, potassium is going to rush outside. Potassium is going to end up rushing outside. So as the potassium rushes outside, what do you think is going to happen to the charges? Is it going to keep going towards the positive? Is it going to stop at 0, or is it going to keep going towards the negative? You're right, it's going to keep going towards the negative. So the point here is to briefly convert the outside to the positive, or the outside to the negative, the inside to the positive, and then revert it back towards the resting membrane potential. So now let's take a look at how these sodium channels work. So sodium channel, when the sodium channel is closed, here the sodium channel is closed. If you see the gate is closed, sodium channels, which are these blue dots, remain outside. When the sodium channel opens up, the blue dots, which are the sodium ions, come inside. The inside becomes positive, the outside becomes negative. When the sodium gates close, they're closed up, the inside, the sodium ions remain on the inside. The inside's still positive, the outside is negative. Now, the sodium ions cannot go back out through here. It's only a one-way street. Sodium ions can come in through the sodium channel, but they cannot go out through the sodium channel. Please note this point down. Sodium channels and potassium channels are a one-way street. Sodium can come in through the sodium channel, but it cannot go out through the sodium channel. Potassium can go out through the potassium channel, but it cannot come back in through the potassium channel. Then how are we going to maintain homeostasis? Enter the sodium-potassium pump. That is how we are going to maintain homeostasis. So the sodium-potassium pump is a pump that is a transmembrane protein. So this is the sodium-potassium pump right here. So sodium typically hangs out in the extracellular fluid right here. This is inside. So this is the cytoplasm right here. Now sodium cannot go back out through the sodium channel, but it has to go back out somehow, right? So it goes back out through, the only way that sodium can go back out is through the sodium-potassium pump. Now once potassium goes back out through the potassium channel, it can't come back in through it. The only way potassium can come back in is through the sodium-potassium pump. Important point to note is that for every 3 potassiums that go out through the sodium-potassium pump, the sodium-potassium pump can only bring in 2 potassiums. So 3 sodiums go out, 2 potassiums come in. So that is how the sodium-potassium pump goes in. And this pump is an active process, so it consumes quite a bit of ATP to work. Now, this is an interesting graph. Now, this conversion of charges happens all the way through the length of the axon. So this happens right here. So the negative 70 millivolts. So on the x-axis here you have time. On the y-axis here you have the membrane potential in millivolts. So this graph does a really good job of explaining what is happening. So I'm going to try to break this graph down and explain how graded potential and action potentials work. So negative 70 is your resting membrane potential. So this is where your neuron is being stimulated. Before this, the neuron is at rest. The sodium is hanging on outside, the potassium is hanging out on the inside, the neuron is at rest. So something happens. You touch a pin and you have that unpleasant sensation, right? So at this point, time goes on, and at this point, at point number 1, is when you touch the pin. And your sodium gates end up opening. As the sodium gushes in, your resting membrane potential, the charge increases as sodium is gushing inside your neuron. Your charge increases from negative 70, it goes to negative 55. Once it reaches negative 55, it is a given, negative 55 millivolts, it is a given. So this is where graded potential, from negative 70 to negative 55 is where graded potential is taking place. Once it is at negative 55, it means that an action potential is taking place. If a stimulus can change your resting membrane potential from negative 70 millivolts to negative 55 millivolts, it means that an action potential is taking place. Therefore, negative 55 millivolts is called a threshold of excitation. It means that an action potential is going to take place. Now this is when the graded potential has reached your axon hillock and the sodium gates at your axon hillock have opened up. Now sodium is gushing inside your neuron at the axon hillock, and sodium is gushing in, Your resting membrane potential, the membrane potential is getting higher and higher and higher and higher. From negative 70, it has now gone up to 0. It's getting into the positive, and it has reached, there's been a change. There's so much sodium entering the neuron that the membrane potential has changed from negative 70 to a plus 30 millivolts. So the membrane potential of plus 30 millivolts is called the peak action potential. So at this point, there is so much sodium inside the neuron that if you hook it up to a voltage meter, it is going to read a plus 30 millivolts. Now at this point, once it reaches plus 30 millivolts, all of the sodium gates have closed. Sodium cannot exit the cell. Potassium gates are going to open up. Once the potassium gates open up, potassium is going to exit the cell. As potassium starts exiting the cell, your membrane potential is going to start decreasing. So now it's going back down towards 0. It's going back down towards 0. The positivity inside the cell is decreasing. It's decreasing. It's going back down towards negative. It's going back down towards negative. It's gone back down to negative 55. It's gone back down to resting membrane potential. So there's been so much potassium exited out of your neuron, and the sodium-potassium pump is doing its job, that your resting membrane potential has come back down to negative 70 millivolts. However, something else is taking place. Potassium gates have not closed down. Your neuron is tired. Essentially, that's what's happened. Your neuron is tired. Your potassium gates have not closed down, and instead of shutting off the potassium gates, they have remained open. More than normal amount of potassium is going out of the cell. As a result, you're losing more potassium than normal. And instead of stopping at -70, your resting membrane potential is going down past 70. So it's going down to -75, -80. Now, when it goes past -80, it is called hyperpolarization. So let's talk about these terms. So from resting membrane potential, when it goes up, it is called depolarization. When membrane potential goes down, it is called repolarization. So membrane potential going up is called depolarization. Membrane potential going down is called repolarization. Membrane potential going past resting membrane potential is called hyperpolarization. So here's how action potential is taking place. Now this neuron, if you look at it, is unmyelinated. You are not seeing any myelin on this particular axon. So once it reaches the axon hillock, which is in this area, this part has gotten depolarized. So this area has temporarily gotten a positive charge and therefore become depolarized. Now once this happens, this area becomes repolarized, and as this area is repolarizing, the temporary reversal of charges moves on here. Once this becomes depolarized, the reversal of charges moves on here, and the temporary reversal of charges moves down all the way down the length of the axon until it reaches the bump at the end of the axon terminal, which is called the synaptic end bulb. Now let me go back here. Now, the way the action potential travels in a myelinated versus an unmyelinated axon is slightly different. You would think the signal travels faster in an unmyelinated axon, but that is not true. The signal actually travels faster in a myelinated axon because the signal jumps from a node of Ranvier to a node of Ranvier. We will talk much more in detail about it in the next part of the lecture. So what happens to this action potential when it reaches the synaptic end bulb? The action potential cannot jump the synaptic cleft. You might recall the term synaptic cleft from our neuromuscular junction. So when the nerve impulse comes down the axon terminal and it reaches that bump, the bump is referred to as the synaptic end bulb. So the reversal of charges travels down in this direction, reaches the synaptic end bulb, and this is where it triggers something. It triggers the opening of a transmembrane protein, which is also an ion gate. But it's not an ion gate for sodium or potassium. It is an ion gate for calcium ions. Once the calcium ion gates are opened, it triggers the influx of calcium ions. Once calcium gushes inside of the neuron, it triggers the exocytosis of certain secretory vesicles. Now the neuron synthesizes neurotransmitters. Now neurotransmitters are synthesized They're proteins which are synthesized and secreted by neurons. One example of a neurotransmitter is acetylcholine. It is abbreviated to ACH. And it is one example of a neurotransmitter. In part 3 of the lecture, we're going to look at different types of neurotransmitters. So acetylcholine is the most common type of neurotransmitter. So in this illustration, these bubbles with the red beads inside it are vesicles containing the stored acetylcholine. So as soon as there is an influx of calcium ions inside of the cell, which are these purple dots, the exocytosis of acetylcholine is initiated. So what you're seeing here is as soon as calcium comes in, exocytosis of acetylcholine is taking place. So these red beads are being exocytosed into this teeny tiny gap between neuron 1 and neuron 2. This is neuron 1, this is neuron 2. So there's a teeny tiny gap between neuron 1 and neuron 2, and that is referred to as the synaptic cleft. So the acetylcholine is being released into the synaptic cleft. Now, as soon as acetylcholine goes into the synaptic cleft, it is going to go and bind to sodium ion channels. So these green transmembrane proteins here that I'm circling right now, let me actually go ahead and change the color. Whoops. Let me go ahead and change the color for the pen so it's not as confusing. So these are sodium ion channels on the postsynaptic neuron, aka neuron 2. Now, as soon as acetylcholine binds to the postsynaptic neuron, it is going to open up these sodium ion channels. Can everyone see how it's opening up the sodium ion channels right here? Now, sodium is hanging out in the synaptic cleft. Sodium, sodium, sodium. Green beads are sodium. So sodium is hanging out in the synaptic cleft. So as soon as the sodium gates are opened, sodium is going to gush into neuron 2, aka the postsynaptic neuron. And as soon as there is an influx of sodium into the postsynaptic neuron, the postsynaptic neuron is going to get depolarized, and the story of depolarization and repolarization is going to begin in neuron 2, and the same process is going to begin in neuron 2 again. So in the next part of the chapter, we are going to talk about how this process takes place in myelinated neurons versus unmyelinated neurons. We're going to talk about the different types of neurotransmitters and how external substances such as drugs, medications affect these neurotransmitters.